coming up next is a company and a person that more than a few of you have been excited about in the chat, in Benzinga Pro chat over the last couple months, but also in the YouTube chat today. So to our next presenter, no pressure or anything, but people are excited. And let's bring them on now. Nano Veracities, it's ticker NNVC, and we have the president and chairman here, Anil Diwan. Anil, how are you?
Good. How are you?
I'm doing very well. It's very good to see you. I will let you take it away, sir.
So my name is Anil Diwan. I'm the president and chairman of NanoViricides. Today, I'm going to give you, okay, let me try to get the share screen completed. What happened to that?
There is stop screen.
Okay.
I'm looking. I'm trying to give you some tips here.
Got it. No, I came to the wrong screen. Escape. I'll have to share screen. Stop screen. Sorry.
Oh, good. Yeah. Usually, if you stop sharing your screen and then try it again, it'll kind of shake something loose. Let's see if we can get that working here.
Yeah, it was this one behind. That I have to go.
I'll let you know when I see it pop back on. So I see it. I see it backstage right now. Ooh, I think whatever you just did there might work. There we are. I see it now. Have at it, Anil.
So,
thank you.
Today I'm going to talk about the exciting advances that we have made in developing a pan-coronavirus broad-spectrum nanomedicines against the SARS-CoV-2 virus and its variants. So these are drugs that we have developed and designed to develop that the virus will not be able to escape, and explain to that why that is important and how we achieved that. We have two different drug candidates now. NVCoV2 is the nanomedicine polymeric micelle nanomedicine. That's our standard approach that we have been working on, that we have called first generation nanomedicines or nanovericides coming out of the company. And NVCoV2R, which is part of our second generation, where we take advantage of the ability of our nanomedicines to deliver additional drugs by encapsulating them. So in the case of NVCoV2R, we are encapsulating remdesivir, which is the well-known antiviral drug. It doesn't work that well in the clinic yet, but with this, we believe that it will be substantially superior. So I'll get to that as well. Okay. Next slide is our disclosure statement. We are a public company, so I am required to tell you that I might be making statements that are not accurate or are far-reaching. I am a scientist, so I do make statements that are far-reaching, and you have to be aware of that, obviously. Most of the presentation is factual, but our expectations are usually what we designed for. They are usually far beyond what people's grasp is even today. So the presentation layout, I'm going to work through SARS-CoV-2 therapeutics development first. The lead drug candidates against SARS-CoV-2, I'll describe them, and then how our strategy works so that the virus would not escape our drugs. Our platform technology, CGMP. We are one of the few companies with CGMP capable manufacturing, R&D, and nanomedicine characterization facilities all in one site in our Shelton campus. That CGMP manufacturing capability is a multi-kilogram capability. So it is sufficient, more than sufficient for clinical trials. And that was our objective. That means that we don't have the stall caused by transfer of technology for manufacturing. So when we say we have done it and we have already done kilogram quantities, that means we are ready for manufacture for clinical as well. The other drug candidate that we were working on previous to switching gears to COVID-19 because of the pandemic is the shingles rash indication. It's a skin cream that was ready to be going into clinical trials. In fact, we were in talks with the clinical trial site when the pandemic hit, in January of 2019. And so very quickly we switched gears. We saw that it was going to become a major pandemic, even when there were no cases yet in the United States. And we started working on it. We had some head start because of our prior work on coronaviruses. And so we were able to develop many candidates by June. And then we decided to focus on a few of them. And then eventually in October, we focused on the candidates that we are working on now. So it's been very, very rapid development. And as you know, the vaccine development was more rapid than ours, and antibody development was more rapid than ours. And that's simply because we didn't have enough staff and we didn't have the kind of funding that went into those programs. Our program can be far faster than even antibody development programs. So what we are doing, the SARS-CoV-2, you see the picture of the coronavirus on the side. What is going on is that it's costing economies trillions of dollars. There is no end in sight, despite vaccines, because variants are fueling new waves. And every time there is a variant, it has escaped some way against the existing drugs, mainly the antibodies and the vaccines. And so we are currently at a stage where we are fighting a constant battle. The booster shots are not going to be the panacea. That helps, but there are a lot of scientific reasons why that is kind of an iffy approach to use. But I'm not going to get into that. I'm not a vaccinologist. I'm not really an immunologist. Whatever I learned is by reading and understanding. So I don't try to get into those things. I will try to focus on our stuff right now. So what is NVCoV2? It is a nanomachine like our standard NanoViricide technology, where we develop a polymeric micelle where the lipid is inside those purple and blue bands that you see inside. That's lipids. And then there is a core, a green core that you see, and a yellow core. Those are hydrophilic or water-loving shell, which is made of polyethylene glycol, PEG. PEG is a very standard magic molecule. It is considered in biotechnology that it protects drugs from getting taken up by the clearance system of the body, RES, reticuloendothelial system, and others. So it has been a magic molecule. And we use that as the backbone of the polymer in the polymeric micelle making. In addition, the great change here compared to other people's approach is that we have those lipids are pendant, meaning they're hanging from the shell, the hydrophilic shell. And what that gives is, it gives this material ability to shapeshift. And that's the important thing. So what happens, if you see that diagram, it's showing you stage 1, the nanomicelle binds to the virus. Stage 2, it starts shapeshifting. It's inside lipid and the lipidic coat of the virus. They start merging together. Stage 3, you see the virus is getting coated. The virus eventually gets fully coated, and then it cannot bind to the cell, or it cannot infect the cell. So that's the approach that we have been using. We have shown that in electron micrography with certain viruses that it actually does happen this way, and probably it goes even beyond that, in that the S protein or the glycoproteins that the virus needs for binding get actually uprooted from the virus surface. So what is NVCoV2R, the remdesivir containing thing? We believe it is a potential cure, and I'll tell you why. You can see the cartoons at the bottom. The one on the left shows you what are the 2 cycles of replication and reinfection that continue to propagate the virus. So reinfection means a free virus in the fluid outside the cell binds to the cell surface receptor S2, goes inside the cell, and then the second part of the life cycle, replication of the virus, generation of the proteins, assembly of the proteins, and egress. Those take place, and that produces the progeny. So in the case of NVCoV2, we are blocking the reinfection cycle. You can see it on the right side. The circle and the green is the blocked reinfection cycle, because the virus is blocked. However, some viruses will escape, and they will cause some residual reinfection in some cells. That is blocked now by remdesivir, which is efficient at blocking these kind of things. The inside replication, it binds to, it acts against the transcriptase, the protein, the enzyme of the virus that causes elongation of the RNA chain of the virus. It blocks that one. So because of that, it's fairly efficient. It's very efficient in cell cultures. It's not that great in the human body or animal bodies. And I'll come to that later, why our approach of encapsulating NVCoV, the remdesivir into NVCoV2 to make NVCoV2R takes care of that problem. So now, because of that, now you have plenty of remdesivir available. It goes into the cells, and that causes blocks, second block. So that's where I have done this XX second block. and there is practically no progeny that could be generated in that situation. So if you have a highly effective viral load reduction like this, then you probably have a cure. From other viral diseases, it has generally been said in the scientific literature or in the scientific opinion that if you get 3 logs or 4 logs reduction, you pretty much have a cure for the viral infection. In the case of coronaviruses, the current antibodies, even in the clinical trials, which means they were against the same strain that was infecting people as the antibodies were raised against, even in those clinical trials, they were getting about only half log reduction of probably up to 0.7, 0.8 logs in the best cases. So they are not really that good. They were very weak to begin with. With each variant, their efficacy decreases with each effective variant that goes through. And we have had now 3 or 4 different cycles or variant waves. So as you can see, the antibodies and vaccines, they are decreasing in their effectiveness. This approach that we have does not suffer from that problem. And that is why we are able to generate something that we believe is a potential cure. So with the NV-CoV-2 alone, we have seen very strong viral load reduction in animal studies. With the NV-CoV-2R, it is even better. So that supports what I'm showing here in the cartoon. Where we are with these studies and the developments, we have shown that NV-CoV-2 and NV-CoV-2R are both broad-spectrum anti-coronavirus agents. We did that using cell culture studies where we used at least 2 different coronaviruses that are not the same as SARS-CoV-2, but one of them is a close cousin of SARS-CoV-2. It binds to the same cell surface receptor as SARS-CoV-2 does. Another one binds to a different one. And against both of them, our drugs were highly effective. Remdesivir is already known to be broad spectrum against many viruses, not just against coronaviruses in cell cultures. Okay. Remdesivir has been relatively less effective in animal studies and in human studies compared to what you would expect from the efficacy in cell cultures. And that I'll get to that later. So substantially less cellular toxicity also we have seen compared to remdesivir. It's not just the activity, effectiveness, it's also toxicity. In the case of remdesivir, we enter into a dose-limiting toxicity regime with the current formulation if you go double the dosing. And that's not good, obviously. So we have been able to, our drugs, both the one with the remdesivir encapsulated, even that one is substantially less toxic than remdesivir. And that's very important. So again, these are effective against unrelated coronaviruses. And I already told you about that. So I'll go to the next slide. We also showed that they are effective in animal studies. And these are with intact, unchanged animals, unlike what was done with development of remdesivir and molnupiravir, where they used genetically modified animals, because of which there is no metabolism or minimal metabolism of the drugs. And so you get false promises of high activity. We didn't do that. The rats metabolize drugs at much faster rate than humans. So that is a given. So this would reflect even lower activity than what you should see in humans. And you can see the data that we have. This is a lung infection of Sprague-Dawley rats with 10⁴ particles, 10,000 particles of NL63, which binds to the ACE2 receptor, same receptor as SARS-CoV-2. So it's a perfectly valid model, surrogate model for that. And with that, when you do not treat the animals or you simply give the injections of the buffer vehicle, the survival was only 5 days. With remdesivir given in the standard format, the 2 infusions 1 day and followed by 1 infusion every day, they survived only 7.5 days. With NV-CoV-2, our nanomicelle polymeric micelle medicine, with a high dose of that, they survived 14 days. And with NV-CoV-2R, with a medium dose, which means it was about half the amount of NV-CoV-2 was in there, and about exactly the same level of remdesivir was there as in the standard remdesivir therapy. They survived 16 days. That is tremendous. So this is why I say we believe, and we pretty much believe, that this is a potential cure. Obviously, because NVCoV2 is a novel agent, we are going to have to do phase 1 and /2A clinical trials with NVCoV2 itself first, and then enter NVCoV2R into phase 2 clinical trials. So that is simply regulatory mechanisms. We cannot help that. But you can see that the drug development has been very rapid and it has been highly effective. I have not seen this level of efficacies in any of the other drug developments. That does not mean that they don't exist. It simply means that I have not doing deep research into these fields. I'm busy with working on these two drugs and taking them to the clinic. So again, in this study of the infection, also we saw significant safety. There was no general organ toxicity with our drugs. Remdesivir did show certain toxicity type of indications at that level, even at that level. And lung histopathology showed that NVCoV2 and NVCoV2R suppressed any plaque formation quite substantially. So we have completed the GLP safety toxicology studies of NVCoV2. As I was saying, we have to take that one into clinical trials first. So in that one, because of its novel drug, we had to enter the GLP safety toxicology studies. Those were completed around January, I believe, and we got the final quality control results and reports in August. So we are ready with that. We saw no evidence of adverse effects. The neuropulmonary safety pharmacology studies were conducted in rats, and they showed that even at 100 mg per kg, there was no respiratory function affectation. GLP cardiovascular function studies were done in monkeys, NHP monkeys, and that is non-human primates. And IV infusion up to 50 mg per kg did not show any toxicological effects or on cardiac rhythm or any ECG morphology. Typically, that's the level these drugs are studied in these animal studies, in GLP studies. So that is where we stopped at. But non-GLP safety toxicology studies we conducted at much higher levels as well. The non-GLP safety toxicology studies were done for both NVCoV2 and NVCoV2R in rats. The rats dosed at up to 560 mg per kg and a total of 3.4 grams per kg dose, which is huge. NVCoV2 showed no side effects. Rats dosed at up to 309 mg per kg body weight on days 6 days for a total of about 1.9 grams per kg dose of NVCoV2R showed no side effects. Now imagine this is carrying 1/10 of that is remdesivir. If you give remdesivir at the same amount to the rats on the same schedule, you get tremendous side effects. So we are having substantial positive benefits of encapsulating remdesivir in NVCoV2R. No evidence of any adverse side reactions was observed. NVCoV2R and NVCoV2R and vehicle groups, they all tolerated the compounds similarly. Note that remdesivir is not mentioned there. The body fluids and fecal analysis showed no significant differences. Histopathological examination showed no changes. So no changes in organ weights or histology also. So these are very, very safe compounds, and we have not reached the maximum tolerable dose yet in that study. We did another subsequent study in which we did determine the maximum tolerable dose. We are compiling the results as of right now, so I can't present those yet. But those results are ready, and that is the baseline for being able to conduct, design clinical trials for phase 1. So we are all set for all those things.
Hey,
now I'll go to
Anil. Yeah. I wanna give you time for maybe like a question or two. Great. So yes, if you want to do like a quick summary, I know it doesn't look like you're quite done with your slides, but if you want to give like a quick summary, we can hop over for a question or two after.
Yeah, I'm not going to go through all the slides. So okay. I'm going to go through only a couple of them. So I want to go through the novel platform technology, what we are doing. So what we do is we make ligands that look like the cell surface receptor, small chemicals. So this is 3 steps of what typical standard scientific research people go through. They usually will take the receptor portion, soluble portion of the receptor alone, Then they will take the binding portion of the peptide from that. Then they will start trying to do binding studies, trying to develop the technology for being able to come up with small chemicals. Eventually, about 15, 20 years later, they come up with the small chemicals. We start by developing the small chemicals, and we do that in a matter of months, 3 months or so in this situation. So I just wanted to point that out, that
Sure.
how quickly we develop these things. And once we make that, that is the recognition site on our polymeric micelle for the virus. So the virus is going to bind to our micelles in preference to the cell, binding to the cells, because the density of the recognition sites is very high. And then our micelle goes to work, like I showed in the prior slide. The mechanism of SARS-CoV-2 drugs is orthogonal to most drugs except antibodies. Antibodies bind to viruses, but before already talked about them being highly specific, and because of that, variants defeat them pretty readily. We are in a good financial position, 20.5 million cash in hand as of June 30th, end of year, reporting year. I'm writing the 10K right now, so it will be published on October 15th. Yeah, so these numbers are officially considered unaudited. Cash expenditure rate is about 2.2 million per quarter. It will go up when we go into engage into clinical trials, and we are really, really striving to get into clinical trials right now. So that's where we are at. Market cap has been hit very badly, and also we did not get the uptick of the market cap that happened with all the companies that were working in this field, coronavirus field, for various reasons. We were always shy about presenting our results before we had validated those results and things like that. So even now we are doing the same thing. We are validating our results first before we talk about them. So comparable market caps of comparable companies are already reached hundreds of millions, and once they hit a clinical milestone, they have gone into billions of dollars. So as you can see, there is a huge breathing gap for the market cap in our case. So so that's basically forms the investment thesis for the company. It's a platform technology. It's the only technology that we know that completely controls the total life cycle of non-persistent viruses, like in NVCoV2R. We have multiple drug programs that will keep on coming, keep on giving. And we have our own cGMP manufacturing facility to push these programs forward rapidly. And market cap I already talked about. So I will stop my presentation at this point. This is the slide where we have shown that the NVCoV2 type polymeric micelles completely dismantle viruses in electron micrography studies.
Anil, I love that you gave us more. I love that you gave us more perspective on like the science. That's not something that we get with all of these execs that are presenting, and it's cool to really see, you know, some of the images behind the actual science. And I just had a one quick question related to that. So I understood the, you know, binding part, how it surrounds the virus. What happens to the cell after it's basically sealed off?
So basically, that is a good word to use. But in this slide that is in front of you, you see that it is not truly sealed off. It's already actually in the slide, in the top left corner, you see the white area. That is where the lipid of micelle is. And everything starts streaming out from the virus particle. It seems to destroy at least some kinds of virus particles. So
okay, gotcha.
After that, it becomes food to the immune system and clearance systems in the body of foreign particle clearance systems, phagocytosis and things like that. We do not really know all the details that happen at that point, but I believe that that would also aid generation of antibodies against the particular virus strain that the patient was affected with.
Okay, gotcha. And then a quick question here from Drew. Drew's asking for any expected time frame for a next clinical trial. Can you speak to that at all?
I can speak to that. We have been saying that we are ready for getting into clinical trials since December. It has been a challenging time frame because more than 300 clinical trials were started. Altogether, 1,500 clinical trials are going on.
Wow.
Vaccines and different kinds of drugs included. So most of the sites were saturated. It's very difficult to get into those, very difficult to get through the, especially the US FDA regulatory screening until you are associated with NIH or something, which we are not. You know.
Sure.
So we are looking at opportunities outside of the country, and we are making pretty good headways there. As soon as we have something concrete, I'll be able to talk about it.
Gotcha. Okay. Well, it seems like a very important technology. So definitely wishing you the best of luck within the U.S. and maybe outside of the U.S. Thanks for joining us today, Anil Diwan. He is president and chair of Nano Veracities. It's ticker NNVC on the NYSE American. Have a good day, Anil. Thank you.
Have a good day. Thank you for inviting us, and good luck to all the people in the audience. Don't get sick. Even though I say things about vaccines, please do take your vaccine doses. That's the only, other than masks, that is the only approach we have right now.
Love the message, Anil. Thank you for saying that. Wise words of wisdom to conclude your presentation here. Great. All right.